Welcome back to the deep dive. Today, we are really buckling up for a big one. We're diving into the human immune system, specifically what happens when things go wrong, when our defenses mess up. We're distilling the core ideas from the pathophysiology text on disorders of the immune response.
Yeah.
And that covers a huge range, everything from, basic allergies right through to something as devastating as HIV/AIDS.
Yeah.
So our mission here is to try to categorize this, make sense of the mechanics when the immune system goes off the rails. We need to understand the three main ways it can fail: underperforming, leaving us vulnerable, overreacting and causing damage to ourselves, or, and this is crucial, completely losing that ability to tell self from non-self.
Yeah, and what's fascinating here really is that even with all the complexity, almost every problem falls into one of those buckets. You've got immunodeficiencies, the system's just not doing enough.
Right.
Then hypersensitivities, where it's way over the top. And finally, that loss of self-recognition and autoimmunity, plus the kind of acquired collapse you see with HIV.
Okay, let's unpack that. Starting with immunodeficiencies. First off, there's this basic split, right? Primary versus secondary.
That's the fundamental distinction, yeah. Primary immunodeficiencies are genetic. You're born with them, some defect in how immune cells develop or work. They're actually quite rare.
Okay.
Secondary immunodeficiencies, though, they're much more common because you acquire them. Something external causes it. It could be malnutrition, maybe chemotherapy, or the big one, an infection like HIV.
Got it. So born with it versus acquired later. And I guess the type of defense that fails really dictates what kind of trouble you're in. Let's start with B cells. If the antibody system, the humoral system, is weak, what infections are the big worry?
Well, if your B cells aren't working properly, you can't make enough antibodies, or big E. And antibodies are your main weapon against bacteria that live outside our cells, especially those encapsulated ones.
Ah, right.
So people with these defects get hit hard again and again with severe infections from what we call pyogenic bacteria. Think Streptococcus pneumonia, Haemophilus influenza, nasty stuff.
Can you give us a specific example, something that really shows the link between the faulty gene and the disease?
Sure. A classic one is X-linked agammaglobulinemia, or XLA. It's caused by a mutation in the gene for Bruton tyrosine kinase, or BTK.
BTK, okay.
Yeah, and BTK is absolutely critical for B cells to mature. They get stuck at the pre-B cell stage if BTK isn't working. No mature B cells means basically no antibodies that block in maturation. That's the key.
Okay, that makes a lot of sense. Now, if B cell failure is bad, I imagine T cell failure, the cell-mediated side, must be even worse, since T cells kind of run the show.
Oh, absolutely. T cell deficiencies are generally much more severe, more profound. T cells are essential for fighting pathogens that hide inside our cells.
Right.
So a defect there leaves you wide open to viral, fungal, protozoan, and intracellular bacterial infections. The most extreme example is severe combined immunodeficiency disorders, SCID. Here, both T and B cell functions are knocked out because of some shared problem early in development.
And SCID, that's the one you hear about being fatal very early on, unless you get treatment, like a stem cell transplant.
Exactly. Without the T cells coordinating everything, and without effective D cells either, the adaptive immune system is just crippled. A hematopoietic stem cell transplant is often the only hope.
Okay. Now, we shouldn't forget the innate system. What about defects there? Like, if the T and B cells are okay, in theory, but the first responders, the phagocytes, aren't working right.
Yeah, good point. That leads us to disorders of phagocytosis. A key example is chronic granulomatous disease, CGD. In CGD, the phagocytes, neutrophils, macrophages, they can actually engulf the microbes just fine.
Okay, so they eat them.
They eat them, but then
Right.
nothing. They lack the ability to produce the necessary microbicidal oxidants, like superoxide radicals. They can't generate that chemical burst needed to actually kill what they've ingested, so the bugs survive inside the cell.
So they swallow but can't digest, leading to these chronic inflammatory masses, the granulomas. Wow. That really highlights the difference between just being there and being functional.
It does. And just quickly, the complement system, too. Deficiencies there, say in proteins like C1q, C3, or C4, are serious. Complement is vital for killing pathogens and flagging them for destruction. But what's really interesting, maybe counterintuitive, is that these particular complement deficiencies are strongly linked to a higher risk of autoimmune diseases, especially systemic lupus erythematosus, or SLE.
Huh. So a deficiency leads to autoimmunity.
It seems that way, probably related to clearing immune complexes or cellular debris. It shows how interconnected everything is.
All right, now for the flip side, we go from too little defense to way too much. Hypersensitivity. These are basically abnormal, over-the-top immune responses that end up damaging our own tissues. And they're classed into 4 types.
That's the classic system, types I through I. 3 involve antibodies. 1 is purely cell-based. Let's start with type I, immediate hypersensitivity.
The allergy one.
Exactly. This is your typical allergic reaction driven by IgE antibodies. It works in 2 steps. First, exposure sensitizes you. IgE gets made and sticks onto mast cells and basophils. Then, next time you encounter that same allergen...
Wham!
Hum is right. The allergen cross-links the IgE on those cells, triggering them to instantly release a flood of inflammatory mediators like histamine, degranulation.
And that histamine wave causes everything from itchy eyes and runny nose to, well, much worse.
Much worse. The most severe form is anaphylaxis. It's incredibly rapid. Sometimes it's graded like ITEV, with grade the 4th being respiratory and cardiac arrest. It's caused by massive vasodilation, leaky vessels leading to shock, plus airway constriction, all from that immediate IgE trigger.
Scary stuff.
Okay.
Moving to type 2. If type 1 is IgE causing chaos, type 2 is different. The antibodies attack things stuck on cells.
That's the key difference. Type 2 is antibody-mediated or cytotoxic. It uses IgG or IgM antibodies that bind to antigens fixed on our own cell surfaces or in the extracellular matrix. And it causes damage in a couple of main ways. One is straightforward destruction. The antibody tags the cell, and then complement or cytotoxic cells destroy it. Think autoimmune hemolytic anemia, AIHA antibodies stick to red blood cells, marking them for elimination. Or like transfusion reactions or Rh disease in newborns.
But wait, the antibodies don't always destroy things. Sometimes they just interfere.
Exactly. That's the second mechanism. Antibody-mediated cellular dysfunction. Instead of killing the cell, the antibody messes with its function. The classic example is Graves' disease.
Oh, the thyroid one.
Right. Autoantibodies bind to the TSH receptors on thyroid cells, but instead of blocking them or marking the cell for death, they activate the receptor, constantly stimulating the thyroid to pump out excess hormones, so you get hyperthyroidism. The antibody acts like a persistent on switch.
That's wild. An attack that causes overproduction. Okay, so type I is IgE, type 2 is e.g. IgM against fixed targets. What about type 3?
Type 3 is immune complex mediated. Here, the antibodies, again usually IgG or IgM, bind to soluble antigens floating around in the blood or body fluids.
Soluble, so not stuck on a cell.
Correct. They form these little clumps called immune complexes. Think of them as like sticky little antibody-antigen bundles. They circulate around and eventually get trapped in small blood vessels or in places like the kidney glomeruli or joint linings.
And wherever they land, they cause trouble.
Precisely. They deposit in these tissues, activate complement, attract neutrophils, and cause inflammation and damage, particularly vasculitis inflammation of blood vessels. Serum sickness is a classic example, or the Arthus reaction, which is more localized. The damage site depends purely on where those complexes get stuck.
Makes sense. Okay, one more type. Type IV. This one's the odd one out, right? No antibodies involved.
Totally different mechanism. Type IV reactions are cell-mediated, sometimes called delayed type hypersensitivity, or DTH. This is all about T cells, specifically T helper cells activating macrophages, or cytotoxic T cells directly killing targets.
And it's delayed. Why?
Because it takes time for the T cells to migrate to the antigen site, get activated, and recruit other cells. We're talking 24 to 72 hours, sometimes longer, to see the peak reaction. The classic example is allergic contact dermatitis, like the rash you get from poison ivy, or the reaction to a TB skin test. It's T cells doing the damage directly, not antibodies.
Okay, we've seen too little response and too much response. Now let's tackle maybe the most fundamental error. The immune system attacking the body itself. Autoimmunity. How does the system normally know not to do that? What is tolerance?
Immunologic tolerance is that crucial ability to distinguish self from non-self. And it's established and maintained through really sophisticated safety checks. First, there's central tolerance. This happens while T and B cells are developing in the thymus and bone marrow. Any cells that react strongly against self-antigens are normally deleted, basically executed before they can cause trouble.
That quality control.
Exactly, but it's not perfect. Some self-reactive cells inevitably escape. So, we have a backup system, peripheral tolerance. This happens out in the body tissues. It involves mechanisms that inactivate self-reactive cells that escaped central dilution, or uses specialized cells like regulatory T cells, TREGs, to actively suppress them.
Sounds like a multi-layered defense against self-destruction.
Right.
So when tolerance fails, what actually triggers autoimmunity? Is it just bad luck, or are there specific causes?
It's usually a mix of things. Genetics definitely plays a role. Certain genes, especially specific HLA types, make people more susceptible, but you usually need an environmental trigger too.
Like an infection?
Often, yes. And one key mechanism linking infection to autoimmunity is molecular mimicry. This is fascinating. It happens when a microbe, say a bacterium or virus, has antigens that look very similar molecularly speaking to some of our own self-antigens.
So the immune system gears up to fight the infection.
Right, it mounts a perfectly appropriate response against the foreign invader. But because of that molecular resemblance, the antibodies or T cells produced also recognize and attack the similar-looking self-tissue. The response cross-reacts.
Ah, like friendly fire caused by mistaken identity.
Precisely. Rheumatic fever is a prime example. The body makes antibodies against group A strep bacteria, but those same antibodies can cross-react with proteins in the heart valves, causing damage.
Okay, that concept of recognizing self versus non-self ties directly into transplantation, doesn't it? The recipient's immune system sees the donor organ as foreign.
Absolutely. The donor organ has different alloantigens, specifically different HLA molecules, which are like the body's tissue type ID. The recipient's immune system recognizes these as non-self and attacks.
And how quickly that attack happens defines the type of rejection.
Yes, we usually categorize rejection based on timing and mechanism. Hyperacute rejection is almost immediate, within minutes or hours. It's caused by pre-existing antibodies in the recipient that react against the donor antigens, often from previous transfusions or pregnancies. It's essentially a type 2 or 3 hypersensitivity reaction in the graft vessels. The graft usually fails right away.
Wow, on the operating table sometimes. What about slower rejection?
Acute rejection happens days to weeks later. This is primarily T-cell-mediated recipient T cells recognize the foreign HLA on the graft cells and attack them. This is often treatable with immunosuppressive drugs.
Okay.
Then there's chronic rejection. This develops slowly over months or years. It's a more complex, smoldering process involving both antibodies and T cells, leading to gradual fibrosis and narrowing of the graft blood vessels, eventually causing the organ to fail. It's much harder to treat.
And one last twist here. Graft versus host disease, GVHD. This happens mainly with stem cell or bone marrow transplants, and the attack is reversed.
Exactly. In GVHD, it's the donor's immune cells, specifically mature T cells present in the transplanted graft, that recognize the recipient's tissues as foreign.
So the graft attacks the patient.
Yes. The recipient is usually immunocompromised to prevent rejection of the graft, so they can't easily fight off this attack by the graft. It can affect the skin, liver, gut, and be incredibly severe, even fatal.
Okay, let's bring this towards the most significant example of a secondary immunodeficiency. We have to talk about HIV and AIDS. This virus does something uniquely damaging. It targets the very cells that are supposed to lead the immune response.
That's right. HIV, the Human Immunodeficiency Virus, is a retrovirus, and its main target is the CD4 T lymphocyte, also known as the T helper cell. These cells are absolutely central to coordinating almost all aspects of the adaptive immune response.
The quarterback of the immune system.
You could say that. By infecting and destroying these CD4 cells, HIV systematically dismantles the body's ability to defend itself. It leads to a profound immunodeficiency.
And it's called a retrovirus because its genetic material is RNA, not DNA, and it has to work backward.
Precisely. HIV carries its genetic code as RNA. Once inside a host cell, like a CD4 T cell, it uses a special enzyme it brings with it called reverse transcriptase to convert its RNA into double-stranded DNA.
Okay, makes a DNA copy.
Right. Then another viral enzyme, integrase, takes that newly made viral DNA and literally pastes it into the host cell's own DNA into its genome.
Oh, so it becomes part of the person's DNA.
Permanently integrated. That integrated viral DNA is called a provirus. And from then on, whenever the host cell is activated, it doesn't just make its own proteins, it also starts transcribing and translating the viral genes, producing new HIV particles. The cell becomes a virus factory, eventually dying in the process.
That's incredibly insidious.
Yeah.
And this gradual destruction of CD4 cells explains the progression of the disease over time.
Yes, typically over 8 to 12 years without treatment. There's an initial primary infection phase, maybe a few weeks after exposure. People often get flu-like or mono-like symptoms. Virus levels, viral load shoot up, and CD4 counts dip sharply. then recover somewhat as the immune system tries to fight back.
And then it goes quiet for a while.
Mostly. That's the chronic asymptomatic phase, or clinical latency. It can last for years, maybe a median of 10 years. The person often feels fine, but the virus is still replicating, usually at a lower level, and the CD4 count is gradually, relentlessly declining.
Until it hits a critical point.
Exactly. Overt AIDS is diagnosed when the CD4 plus count drops below 200 cells per microliter, or when the person develops certain specific opportunistic infections or cancers that define AIDS. At this stage, the immune system is severely compromised.
And that's when you see those opportunistic infections, things that wouldn't normally harm someone with a healthy immune system.
Correct. Things like pneumocystis pneumonia, Kaposi sarcoma, severe fungal infections, certain lymphomas, they take advantage of the weakened defenses.
And treatment today, HAART, it works by targeting those viral enzymes, like reverse transcriptase.
Yes, HAART stands for Highly Active Antiretroviral Therapy. It's usually a cocktail of drugs that hit different stages of the HIV life cycle. Some block reverse transcriptase, others block integrase, some block the protease enzyme needed for viral assembly, or block the virus from entering cells in the first place. It doesn't cure HIV, but it can suppress viral replication dramatically, allowing the immune system to recover significantly.
And quickly on diagnosis, we screen with ELISA, confirm with Western blot.
Generally, yes. EIA or ELISA tests are used for screening. If positive, a confirmatory test like the Western blot is done. But it's crucial to remember for infants born to HIV-positive mothers, these antibody tests aren't reliable because the baby carries maternal antibodies for months.
Ah, right.
So for infants, you need tests that detect the virus itself, like PCR to look for viral RNA or DNA, hashtag outro. So if we pull back and look at the whole picture from this deep dive, we've really covered the three main ways the immune system can fail, haven't we? We saw immunodeficiencies, just not enough response. We saw hypersensitivities, way too much response, causing damage. And we looked at autoimmunity and HIV, the breakdown of self-tolerance or a targeted destruction of the system itself. Recognizing these big patterns is key.
It really is. And it leaves you with this thought, doesn't it? That the immune system has to walk this incredibly fine line, this knife edge. React too little, and infections overwhelm you. React too much, or against the wrong target, and you get autoimmunity or allergy.
A constant balancing act.
Yeah. And the challenge in treating all these disorders really is trying to somehow nudge that balance back towards the center to restore that precise control without causing new problems. It's incredibly complex. Well, thank you all of you for joining us on this deep dive into the often confusing world of immune response disorders. We hope breaking down these mechanisms of failure, overreaction, and loss of self-tolerance has been helpful. Keep digging into these fascinating connections in pathophysiology.